So this is a molecular genetics FRQ, and to synthesize a polypeptide we need to go from replication to transcription to translation, and to do this we need nucleotides, though sometimes the nucleotides can be deleted or inserted, or they can be substituted like a penguin to a panda. Actually no, just nucleotides. Okay, so here we have a strand of nucleotides and they're grouped into three, so then they can be coded as codons. And each of the codons code for amino acids, which bind together to create polypeptides and proteins and kind of run the show and do stuff in our body. So first in the process we have replication. And in replication we have a helicase enzyme which unzips the DNA polymerase which adds new complementary base, and then we have an RNA primer to get everything going. Next we have transcription to go from DNA to RNA. And we need the RNA polymerase to separate two strands of DNA, transcription factors, which are proteins that help RNA polymerase bind DNA, a promoter, which is start point on DNA, a terminator, which is the end point on DNA, and then the actual part of the DNA that is transcribed is called the transcription unit. So now we have our RNA, which can be translated into polypeptides. So the codons are read by the transfer RNA, or tRNA, which is made of an anticodon and an amino acid on the end. So the amino acids bind together to make polypeptides, which are proteins, which are pretty fantastic, almost like superhero powers. Actually, they can just make you happy and act as enzymes, transport, defense, receptor protein, signaling, motor, and storage, hormonal, and structural functions. And this is, so can nucleotides really have that big of an effect on protein function? Well, if it is a deletion or insertion mutation, it really can. So for deletion of a nucleotide is basically just removed. However, all nucleotides downstream of this deletion is then altered due to the improper grouping. And this can result in premature termination or the protein being non-functional. Similar effect can happen when there is an insertion of a nucleotide. So all nucleotides downstream of this insertion are then improperly grouped. And this can have many changes in the code for the amino acids. These are both known as frame shift mutations. There are also pair substitution mutations, which just means that a nucleotide is substituted. However, there are silent mutations when the nucleotide that is substituted still codes for the same amino acid. As such, when the A is changed to the G, it still codes for leucine. There are also nonsense mutations, which happens when the nucleotide changes, so the codon codes for a stop codon rather than an amino acid, which can cause a lot of other problems as well. And then there are missense mutations in which the nucleotide is changed and it codes for a different amino acid. However, this may or may not have that big an effect on the protein due to the location or the structure. It could be similar to the amino acid that it replaced. So changes in nucleotides can or may not have large effect on protein and function.
Created using Powtoon.